Okay, so this is the lesson 7 group problem. So for the first problem, it was asking about what the composition of each of like the genomes for the different viruses was. And so for the first genome, it had A was 28%, C 22%, G 24, T 0, U 26. So first of all, we know that it is RNA because it has uracil and it doesn't have T, and then we know that it's double-stranded because if you look at the numbers, they're pretty much even, so like A and U are about even, and then we know that it's circular because it's resistant to exonucleases, but it's not resistant to endonucleases, so it's protected because of that circular structure there. Then the genome for virus 2 is SS linear RNA, and it's single-stranded because it's sensitive to both exonucleases and endonucleases. So that's why it's linear, not circular, and then single-stranded, not double, because A and U, there's 34 and 15, so that's not really evenly going together. Then the genome for virus 3 is double-stranded linear DNA, and I've kind of gone over everything in this one, but then it would be DNA there because it doesn't have any uracil in it. And then for virus 4, it's SS circular DNA, and again that's because it has no uracil. It's resistant to exonucleases but sensitive to endonucleases, and it's not even between the base pairs. Then number 2, I'll just read through that. We have topoisomerase is composed of 4 protein subunits and uses ATP to compact bacterial chromosomes, while a nucleosome contains 146 to 147 base pairs of DNA and 8 histone proteins. The DNA double helix is 2 nanometers wide. Adjacent base pairs are separated by 0.34 nanometers in the DNA double helix. Topoisomerase 1 introduces positive supercoiling into DNA and cuts one DNA strand. The origin of E. coli is called orexy. And in the Hershey-Chase experiment, bacteriophage T2 proteins were labeled with a radioactive isotope of sulfur, while bacteriophage T2 DNA was labeled with a radioactive isotope of phosphorus. The sugar in uridine is ribose, while the sugar in deoxythymidine is deoxyribose. And then Abby's going to do number 3.
Okay, so for A, you know that 1 BP equals 0.34 nanometers. 1 nanometers equals 1 times 10 to the negative 9th meters. So you get the amount of base pairs there are, which is going to be chromosome 14 base pairs plus the chromosome 21 base pairs. Going to get you 1.58 times 10 to the 8. Once you multiply times 0.34 times 1 times 10 to the negative 9. And that's going to give you 0.054 meters. For 2, you're going to divide 158 million by 197 BP, which gives you 102,030 nucleosomes. 2 over C, you're going to multiply 2 times 102,030, which will be 1,604,060, which was H2A. And for D, so very similar to A, 158 million, 9.34 nanometers over 1 BP times 1 loop over 30 nanometers, which was 101,790,667 loop domain.
Okay. So for I don't have my sheet with me. Oh well. Okay, so for A we took the 4,410,000 base pairs, and then we multiplied it by 0.34 nanometers over 1 base pair times 0.001 micrometers over nanometer, and then like you can see what canceled there, and then that leaves you with 1,499.4 micrometers, which is the answer for A. And then for B, all you do is you just take that number and you divide it by pi, which gives you the diameter, which is 477.274, and that should say micrometers, and that is
Okay, so for number C, we have a 50,000 of the base pair per each loop. So we divided by total number of the base pair by the one loop. Then we got 88 loop domains, but one of them is stretched. So 87 loop domain, and the total length of that loop domains are times 30 nanometers of it. And then add of them, one of the stretched loop domain. So 50,000 base pair times 0.34 nanometers over 1 BP, and then I got a 19,160 nanometers. Then I converted that to the micrometer by times 0.001 micrometer over 1 nanometer. So I got a 19.16 micrometer. And then for D, I set up as the one nucleosome has a 197 base pair each. So total number of the base pair of this circular chromosome divided by 197 base pair times 8, because one nucleosome has 8 histones. So I got a 179,086 histones. Okay, so number 5.
we just go to your screen.
So number 5, A is the deoxyadenosine, and then B is deoxyadenosine triphosphate, C is cytidine triphosphate, 4 is deoxythymidine monophosphate, E is just the cytidine, and then the last F is uridine. So for first one, the nucleosides are A, E, and F. Nucleotide is B, C, and D. And then which contains a ribose is C, E, and F because it doesn't have a deoxy in front of it. Then deoxyribose attached to it is the A, B, and D. And then molecular part is the pyrimidine is C, D, E, and F.
Alright, and that's it.